We are an interdisciplinary research team that combines expertise from various areas of science and engineering to address key challenges in biomedicine. We study diseases on multiple scales, from molecules and cells to organs. For example, we use optical tweezers to observe how a single molecule of protein or a single molecule of DNA can fold or unfold in a cellular environment. In parallel, we employ circuit topology, a unique modeling technology developed in our group to study the folding of complex proteins, such as those with intrinsically disordered domains. These proteins are involved in neurodegeneration, muscular dystrophy, and cancer. The disordered proteins can form tiny condensate droplets that undergo liquid-solid transition in diseases, a process that we study using advanced optical and force spectroscopic methods. Our cell research is primarily focused on immunomechanics and mechanomedicine. We have developed several single-cell assays which can be used to study mechanical properties of human cells, such as immune cells in cancer or vascular cells affected by Ebola and Dengue. These viruses can disrupt cellular mechanics with fatal consequences for patients. As such, our research is a key step in preparedness against viral diseases with epidemic potential. This involves not only single-cell analysis, but also modeling mechanopathology using organ chip and bioprinting approaches, which open new avenues for drug development.